All right, our next speaker will be Nicholas Brazeau. I apologize, I probably butchered that, from the University of North Carolina. And he'll be talking to us about Plasmodium vivax whole genome sequencing to assess genetic relatedness in polyclonal relapses in Cambodia.
Thank you to the organizers. As I said, my name is Nicholas Brazeau. I'm from the University of North Carolina, working with the UNC IDEAL group. And today I'll be sharing our work on whole genome sequencing of polyclonal Plasmodium vivax relapses in Cambodia. And so a disclaimer from our Army collaborators here. And so the story that I'm going to be telling today starts in 2007 in two studies using a few genetic markers with individuals removed from an endemic to a non-endemic setting, as well as separately individuals in a low-intensity transmission setting, showed that vivax relapses typically had heterologous activation. Heterologous activation can be defined as a different clone seen at the relapse blood stage infection than during the initial infection. And so this is indicated here by the different colors of the clones, or the clones are different colors, and so you see gray at the initial, and then successive relapses in blue and purple. And so from this work and others, a theory of clonal activation arose, that there was an internal clock or an intrinsic factor that triggered reactivation of the relapses. However, recent work using microsatellites and amplicon deep sequencing, very sensitive to the minority clones, has shown that this may be a much more complicated picture with both homologous activation and heterologous activation, as indicated here by the blue and gray clones of the initial infection, as well as in the later successive infections. And so if the initial infection and the hypnozoite were from the same inoculum, the same infectious event, we may expect them to be more related than any two random strains from Plasmodium vivax population. This increased relatedness would result from the fact that recombination happens in the mosquito midgut, creating meiotic siblings. So these heterologous clones, in expectations, some of them may be meiotic siblings. And so although they differ at specific genetic loci on the whole genome scale, they should be very similar, or may be very similar. And so the 2 points that I wanted to take forward are that in an endemic setting, we expect vivax to be very multiclonal, and successive relapses should show increased relatedness or be more related than any two random strains from the population. And so the samples that I'll be talking about today come from a military personnel cohort study from northern Cambodia that were infected with Plasmodium vivax. We'll be focusing on 4 patients that suffered from multiple recurrences. Of note, Primkin was withheld until the end of the study, and it was not given at the beginning of the study. So what I'll be calling moving forward initial infections themselves could actually be relapses. And so this is amplicon data of MSP1, a variable target, hypervariable region, for the 4 patients. Patients are separated by gray or white space here. And on the x-axis, you have each successive episode, with R0 being the initial infection, and sequential numbering of the successive infection. On the y-axis is proportion of haplotypes seen within that infectious episode, with the different colors representing a different haplotype in the proportion. And then at the top is the multiplicity of infection estimate, which is just the number of strains, the number of different haplotypes we saw based on this MSP1 amplicon data. And as an aside, this haplotype reconstruction was performed using SeqDeep, which is a fantastic program by Nick Hathaway, and I really encourage you to go and talk to him if you're as excited about amplicon reconstruction and haplotype reconstruction as I am. And Nick's not paying me to say that, for the record. So as you can see here, a lot of diversity, and this amplicon data might be good for identifying minority clones and having a really high level of clonal detection, but would give a biased or perhaps not holistic view of genomic relatedness, since we're only looking at a single target. And so the aims of our study were to see if we could determine the multiplicity infection or recapitulate it on a whole genome scale that we saw with the amplicon data, as well as to assess identify genetic relatedness among the successive episodes. And so this is a really busy slide, but the 2 main points that I want you to take away are we took 70 previously published whole genome sequences from the same region and nearly the same time period that were previously published by our group, combined those with our 12 samples across the 4 patients, joint variant called, hard filtered, and ended up with about 4,000 SNPs that were spread throughout the genome. And so these approximately 4,000 SNPs are what I'll be using throughout the rest of the analyses here. And so, again, high multiplicity infection was shown by whole genome analysis. To orient you, on the y-axis, again, is samples, and patients are blocked with either white space or gray space. On the left is a plot of FWS, which is a measure of internal heterozygosity, with 0.95 or greater being a cutoff that indicates monoclonality. So as indicated here, it looks like all of our samples are multiclonal. In the center is the MSP1 amplicon data that we've been previously discussing. And on the right is multiplicity infection as determined by the real McCoyle, which uses a Bayesian method to infer a discrete count of multiplicity of infection. So again, a high level of multiplicity infection, although there doesn't seem to be any clear pattern among successive episodes of it, multiplicity infection decreasing or increasing within a patient. And so, as you can see here on the x-axis, which is multiplicity infection estimates by SeqDeep, on the y-axis by the real McCoyle, we're getting different results of the number of the multiplicity infection depending on the methodological approach we used. And we think this was largely due to differences in depth of coverage, perhaps using too stringent a SNP filtering. For example, there were 0 SNPs that passed in that MSP1 region that we've been talking about with the amplicon data. But also the noted realization that there is allelic dropout with hybrid capture, which was used in selective whole genome capture, or selective whole genome amplification. So this really kind of emphasized the point that it depends on the goal of your analysis, whether it's for clonal detection in depth, or if you want to look at breadth and perhaps genetic relatedness, that should really drive your wet lab approach. So moving on to AIM2, being able to identify genetic relatedness in these successive episodes. Hopefully I've convinced you that these samples are polyclonal. And having polyclonality or high multiplicity infection and doing genetic relatedness analyses in these samples becomes complicated, because what happens is if in a single sample you have multiple strains, the strains may have discordant alleles at specific sites. So this results in heterozygote calls or discordant calls at those specific loci. And so traditional approaches that people have taken to handle these het calls are either to call the major allele or to just mask those regions. So I've taken the former approach of just calling the major allele here. And within patients, there was some indication of clustering by principal component analysis. If I can convince you from multiple perspectives and tilt your head sideways perhaps, there is at least some indication of clustering, which was somewhat recapitulated by our structure analysis. However, this completely, was essentially, the resolution was lost when we took this onto a population level. So this is on the background of the 70 Cambodia samples that I mentioned earlier. And you'll see that we lost some of the fine-tuned resolution of within patient clustering that we hope to see. And so we realized that, well, we thought this might be due to the fact that we aren't using the heterozygote calls to our advantage. We want to use all the information in the genome to actually be able to infer this fine-scale clustering that we're going after. So under the guidance of Bob Verity, we set out to develop a program that used identity, or that estimated identity by descent from unphased polyclonal samples. And this builds on work from a hidden Markov model framework that other groups have worked on, but we're trying to extend it here to assess any level of polyclonality, not just 1 or 2, as well as to be able to infer multiple parameters associated with identity by descent. And so we performed a simulation for our model, and you can see on the left there, what we've done is I've taken 2 samples, and each sample has 3 strains within them, and I've made between samples specific regions of the strains identical, as indicated by the yellow, orange, and blue boxes. And if you move over to the plot on the right, on the y-axis is the number of genotypes shared, 0, 1, or 2, and the x-axis is moving along the genome. So at regions where there's 2 genomes shared by the 2 samples, or 2 genotypes shared by the 2 samples, we'd like the model to infer that information, as well as to be able to infer other parameters. And so, yeah, to draw your attention, this is what I'm talking about when I say 2 genotypes shared in the model inferring the 2 different genotypes. Okay. And so moving to the full model simulation, in the first plot there, again, on the y-axis, you have pairwise comparison between strains, with green blocks being identity by descent regions or identical regions. In the second plot, you have what the variant call file would look like if you were to just variant call these 2 samples traditionally, with heterozygote calls indicated in yellow. And I think from that variant call file, or at least I have a really hard time seeing any kind of identity by descent between those 2 samples. However, if you move to the third plot, which is our posterior probability distributions as inferred by poly-IBD, our model, with the yellow indicating a higher probability that that is the number of genotypes shared, and you look at the white line, which is truth from our simulation, if our model is working, you would expect to see the yellow, the higher probabilities tracking the white line. So to just hammer this home, zooming in on a specific region here. So if you look vertically now, you would see at this specific couple of loci that there are three strains. The three strains within the two samples have an identical region, the resulting variant call file, and our model is inferring that at that region there are actually three genotypes shared. And again, that's truth by the white line there. So we were really excited to see this. Our model seems to be working really well. We're being able to infer the number of genotypes shared between two samples in a highly polyclonal situation without having to do any phasing or anything like that. And so we took this to the poly-IBD, or we applied poly-IBD to a single patient, and looking at this patient, we were able to see that there is signal of large chunks of identity by descent shared, and this is just across chromosome 1, and it's indicative that there may be, as we predicted, successive relapses are highly related. This is still under development and something that we're still working on with optimization of further parameter estimation. We also need to improve the speed of this program to make it actually really applicable. And so we're also working on estimating the bias and the power of the model and our limits of detection. So then, in summary, for the second aim, traditional approaches that don't actually leverage the heterozygote calls may be underestimating genetic relatedness, and future work is going to be aiming to use poly-IBD to infer genetic relatedness and identify meiotic siblings among these successive infections. And so I'll give us half marks on our second aim as work is still ongoing. And with that, I need to thank a number of people, primarily my advisors, Jonathan Giuliano and Steve Meshnick, as well as the entire Ideal at UNC group, Jessica Lynn, who conceptualized this project and let me work on it, the Carleton group, AFRAMS. A special thanks to Bob Verity for his help with poly-IBD and his guidance and mentorship. And I need to thank the UNC MD PhD program for their support. And with that, I'll take any questions.
We have time for a couple questions.
That was very nice. I just wanted to make a comment too. So we played around with this too, and one thing one might eventually need to do to clean up the data is to have two different types of SNPs, and because what I'm seeing is, there's these genes that seem to be recombining at very high rates, possibly Vir genes, PFEP1s, and they sometimes have different behaviors, than the core SNPs in housekeeping genes. So if you were to separate those different types of SNPs based on their functional classification, you might get even cleaner results.
Okay.
You thought about that. Anyway.
Thank you. No, that's an excellent point, and I think
all variants are not created equal, I would say.
Absolutely, yeah. And I mean, we've played around with the alignment parameters even to try to align to those hypervariable regions, because sometimes, you have multi-mappers with the reads and things like that. So I think we throw out a lot of those just because, like you said, they might be mapping to multiple places and have lower scores, and so they might be valuable. Thank you.
Anyone else? Okay, thank you very much.
Thank you.